Welcome back, class. Mr. Rasmussen here. In this lesson, we're going to be talking about non-Mendelian genetics. What we've been talking about before now has all been genetics rules that Mendel has come up with. So we've been talking about the law of segregation, the law of independent assortment. We've been talking about dominant and recessive and how genes are carried from parent to offspring. So if we're looking at pea plants, and we know that the violet flower over here is dominant, and we know that the white flower is recessive. We know that if we cross them, we'll get all violet flowers, depending upon their genotypes. So that's Mendelian genetics. But the bad news of this is sometimes life is just not this easy, and genetics is the same way. There's a lot more going on than Mendel realized, especially in human beings. Our genetics are a lot more complicated. For instance, what about if we look at hair color? Which hair color is dominant? Is brown dominant? Is red hair color dominant? Is blonde hair color dominant? Which shade of brown is most dominant? So it's hard. We have a lot of different genetics going on there with hair. It's not as easy as simple, well, brown hair is dominant over blonde, and therefore if we cross this, we'll get this. It's not that easy. There's a lot of different shades of brown. Another example would be human height. If we take a look over here, we have one human being down here who's really short, and one human being over here is quite tall. So which one's dominant? Is tall dominant? Is short dominant? We don't really know. There's a lot more going on than just dominant and recessive in Mendel's two laws. So that's what we're going to talk about, some exceptions to these rules, some laws that don't quite follow exactly the way Mendel said they would. One example is incomplete dominance, and its name actually tells us what it is. In this case, the dominant trait is not going to completely dominate over. It's not going to completely hide the dominant trait. A great example of this is the snapdragon. It's a type of flower, and its flowers come in white, and they come in crimson. So there's the white snapdragon and this very pretty crimson flower down here at the snapdragon. And they come in these two forms, and the crimson form is dominant and the white form is recessive. But the problem is when we breed this, when we do the cross, if we take two dominant and we cross them with two recessive, right? Oh, it's hiding my letters here. If we cross them with two recessive, right? We end up with something different. We end up getting a pink flower. So that's not exactly the way Mendel said it would work. He should say that the dominant flower, the red, should hide the white flower. It should mask it. But in this case, the pink flower is shining through. Well, what's happening is something called incomplete dominance. The crimson gene is not completely dominant over the white gene. And so when they're together, you're seeing both of them. Let's look at an example. If we look at this example here, here's our crimson flower over here. And here's a white flower. So we know that the crimson is homozygous dominant, and the white is homozygous recessive. But what happens in any time we see that heterozygous, anytime we see both genes, the dominant gene, that big R, is not completely masking the little r. So we're still seeing part of the white, and it shows up as purple. So in incomplete dominance, the heterozygous, the big R, little r, the heterozygous, looks different from both of the homozygous, dominant and recessive. And that's because you're seeing both traits. The dominant is not completely dominating. That's a really easy one to remember, the incomplete dominance, because its name really says it all. Its name really says it all. Okay, let's look at the next example. Let's just look real quick at sex chromosomes. So to make sense of sex-linked traits, let's look at the sex chromosomes. And over here, we have a beautiful image of the X chromosome and the Y chromosome laid out side by side. And one of the main things you notice is that the X chromosome is quite a bit bigger. It's by far larger than the Y chromosome. The Y chromosome is really small. And here's another little picture that kind of shows that. The X chromosome over here, this X chromosome, is huge. It contains a ton of genes. There's a lot of genes on this chromosome, a lot of different traits. But the Y chromosome is really small, and it doesn't contain many genes at all. very few. So which means, so if we look at males, or let's look at the difference between males and females again. We know that females have two X chromosomes. Okay? And we know that males have an X and a Y. Now, if there's a gene found on the X chromosome, females are going to get two copies of it. Two copies of it. Because they have two X chromosomes, so if that gene is found, maybe that gene is right here on the X chromosome, well, females are going to get two of them, because they have two X chromosomes. But males, on the other hand, are only going to get one, because the Y doesn't contain it. Which means whatever the male gets is what you see. So if the male has the dominant trait, you see the dominant. If the male has the recessive trait, you see the recessive trait. Which means that in sex-linked traits, or traits that are found on the X chromosome, it's a lot easier for males to get the recessive version of that gene. Simply because whatever they get, they show. So if they have only one little N, they don't get a second chance to mask it with the dominant. So let's look at some examples of sex-linked traits. Here's an example, and this would be a good example. This is an X-linked, so it's found on the X chromosome, and it's recessive. So let's look at a good example of this would be colorblind. Okay, so color blindness works this way. So over here we have an unaffected father, which means he has the dominant. So he's going to be X. We'll use B for color blindness. So he's X, big B, Y. Okay, so he doesn't have that trait. He has the unaffected or the dominant version. The carrier mother has two X's. So she's going to be X, big B, but she's a carrier, so which means she has the trait hidden. So she's X big B, X little b. So here's her carrier. She has the gene hidden, so she's the heterozygous version. Over here in her children, when we do the Punnett square for this, we notice that we get an unaffected son. So if we take this Y from dad and this X from mom, we get an unaffected son. If we take this X from dad and this X from mom, we get an unaffected daughter that's not a carrier. If we take the X from dad and the second X, we get a carrier daughter. And then if we take the little x, so this one over here ends up getting mom's little x and dad's y. This one we end up having a colorblind son right here. So in this thing, it's a lot easier. You notice that the girls, we don't have any female children with the trait. Sure, one of them's a carrier, but none of them actually have the trait. But in this case, 50% of the males end up having the trait. And that's just because it's easier for males to get it because it's only found on the X chromosome. If we look at an example problem, and this is where we'll stop today, we'll just go through the example problem we'll be done. It says, a normal dominant male has children with a woman who is a carrier for colorblindness. So this is the same problem we just saw. What is the chance that they will have a male child who is colorblind? So if we were going to do the Punnett square for this, we would just first draw out the genotypes. So we're looking at a normal male. So he is X, big B, Y, right? He is a normal male. And we're looking for a woman who is a carrier. So she is X, big B, X, little b, right? And we're going to do the Punnett square cross for this, and this is sort of what we didn't see in the last image. So we'll draw out the Punnett square as best I can using a mouse, which is not amazing. Here's my Punnett square. I'll put dad along this side, so he's X, big B, Y. And then I'll put mom along this side, so she's X, big B, X, little b. And then if I look over here, if I cross these, we notice we get X, X with both big B's. We notice that this one is X, big B, X, little b. This one ends up being X, big B, Y. This one ends up being X, little b, Y. So when we look at this here, we notice that up here, this one here, we have a normal female. 2 X's, 2 big B's, nothing. Normal female. Up here, we notice that we have another normal female. Now, she is a carrier, but she still doesn't have the trait because she has the dominant to mask it. Down here, we have a normal male, and we have a normal female. A normal male, and down here in this box, we have a colorblind male because he only has one little B. So what's the chance that they will get a male child that's colorblind? Well, we don't care about the female children, so we can ignore these up here, and we can just look at the male child, and it's 1/2. So 1/2 of the male children are going to be colorblind, or 50%. 50%. So that's how we look at a sex-linked trait problem. We're just going to look at using the X's and the Y's and making sure we put the genes above them, whether they're dominant or recessive. And that's how sex-linking works. If you have any questions, class, please feel free to ask me. Otherwise, have a good day.